Hello, my name is Asma. I am one of the authors alongside Clarissa, Julia, and Vanessa. Our topic is modeling the relationships between tick abundancy and Lyme disease pathogen. Vector diseases are diseases that are spread through the bite of an arthropod. Arthropods act as vectors for disease and play big roles in spreading diseases. Ticks are often known as vectors. Ticks can spread a pathogen that causes Lyme disease. Vectors are vital for a pathogen's success. Is there a correlation between vector biodiversity and reports of vector diseases? This is the question that is trying to be answered in this project. Biodiversity is a measure of variability of organisms. In this case, it is the variability of vectors that is going to be looked at. Even though the current information provided by study on vector biodiversity is rather limited, some studies seem to agree that there is no significant or direct link between vector biodiversity and reports of diseases. Other studies speculate that there is a link between biodiversity and diversity prevalence. Studies also suggest an indirect link between biodiversity and disease prevalence, whereas biodiversity has a significant effect on vector abundance and therefore disease prevalence. To further investigate on the question, we will look at the biodiversity of ticks and pathogen reports.
So what we did to answer our hypothesis was that we used the data from the NEON portal, which was a collection of different samples from ticks that were collected in Petersham, Massachusetts. We used data that went back as much as 2014 and up to 2020, and we broke it down by year for each group member to tackle. What we looked at was the number of ticks counted each of those years and the different species and genus of ticks as well. Then after that was done, we broke down each year of how many ticks each year were counted that were known to be positive for Lyme disease, which means they would have the bacteria known for it, and then we compared it to the ones that were negative for Lyme disease as well. Once this data was collected, we broke it down into different graphs. One of the graphs was comparing the average nymphs abundance to larva abundance, and then one was in comparison to the number of positive and negative ticks for Lyme disease over those course of all those years. Then, once the weather got better, we went outside to Westfield State and we tried collecting raw data ourselves by using 2 wooden poles and a cloth that were attached to each pole and dragging it over a measured plot, which were like 10 by 10 meters, I believe, to see if we could attempt to collect any ticks. After that, we made an RStudio code to help us with our calculations as seen below. Along with other stuff, we also compared the different temperatures and how many ticks were seen in each temperature. The average temperature for the area that was in Peterson's, Mass was an average of 7.833 Celsius a year. And then we used an SIR model and the appropriate representations for each of these values to see how many of the infected individuals with Lyme disease recovered, and we also did a Shannon index as well in the use for our calculations.
For our results, we created 2 graphs and linear models. The first graph we created represents the average of positive and negative ticks over the years of 2014 to 2018. Positive ticks are shown in red and negative ticks are shown in blue. As you can see, there was a greater abundance of negative ticks versus positive ticks. Our second graph shows average nymph abundance and average larval abundance per month over the years of 2014 to 2018. Average nymph abundance is depicted in blue and average larval abundance is depicted in red. We can see that both average nymph and larval abundance remained low for the months of April leading up to June. In June, nymph abundance peaks and went back down afterward. And the month of August was when larval abundance peaked, and it starts to go down afterward as well. As a result of our linear models, we found no significant statistical relationship. Our first linear model assessed ticks positive for Lyme as a function of nymph and larval abundance. This model yielded a p-value of 0.5415. For the second model, ticks positive for anaplasma were modeled as a function of nymph and larval abundance. This model gave a p-value of 0.7885. With that said, our null hypothesis was accepted. We expect that this was because of our limited data and testing capabilities. A study done in 2014 by Foley and Scott looking at pathogen prevalence in ticks in California communities found that Lyme disease prevalence was linked to tick abundance. And they also found that Lyme disease was mostly prevalent in adult stage ticks. Our study only looked at nymph and larvae, which may have limited our results. Additionally, the environment may not have been ideal. Wood and Lafferty 2013 said that Lyme disease is generally prevalent in rainforests. The data we got from the Harvard forest had overall low abundance of positives. The highest number of positives we observed was 22, and for some months there was no testing at all. So we have some ideas on what could be adjusted for a more reliable future study.
Some future experiments that we would like to do would be that we would like to expand our research area, and we would like to research more ticks to see what the abundance is with the Lyme disease, to see if it's actually as high as what we received with our data. And then we also want to see the tick abundance is rising and also decreasing. So we would also like to be able to physically collect our ticks and test them for Lyme disease and other possible diseases that they may carry, because unfortunately this time around we weren't able to physically go out and collect the data, and when we did, we got nothing for data. And I think part of that had to do was because it was too early in the season to collect anything. But thank you so much.